PSMA6. Proteasome subunit alpha type -6 is a protein that in humans is encoded by the PSMA6 gene. This protein is one of the 17 essential subunits alpha subunits 1,7, constitutive beta subunits 1,7, and inducible subunits including beta-9, beta-2, beta-5 that contributes to the complete assembly of 20th proteasome complex. Structure. Protein expression. The gene PSMA6 encodes a member of the peptidase TONA family, that is a 20S core alpha subunit. A pseudogene has been identified on the Y chromosome. The gene has 8 exons and locates at chromosome band 1413. The human protein proteasome subunit alpha type -6 is also known as 20S proteasome subunit alpha-1 based on systematic nomenclature. The protein is 27 kDa in size and composed of 246 amino acids. The calculated theoretical pi isoelectric point of this protein is 6.35. Complex assembly. The proteasome is a multicatalytic proteinase complex with a highly ordered 20S core structure. This barrel-shaped core structure is composed of 4 axially stacked rings of 28 non-identical subunits. The 2 end rings are each formed by 7 alpha subunits, and the 2 central rings are each formed by 7 beta subunits. 3 beta subunits beta-1, beta-2, and beta-5 each contains a proteolytic active site and has distinct substrate preferences. Proteasomes are distributed throughout eukaryotic cells at a high concentration and cleave peptides in an ATP/ubiquitin-dependent process in a non-lysosomal pathway. Function. Crystal structures of isolated 20S proteasome complex demonstrate that the 2 rings of beta subunits form a proteolytic chamber and maintain all their active sites of proteolysis within the chamber. Concomitantly, the rings of alpha subunits form the entrance for substrates entering the proteolytic chamber. In an inactivated 20S proteasome complex, the gate into the internal proteolytic chamber are guarded by the N-terminal tails of specific alpha subunit. The proteolytic capacity of 20S core particle Cp can be activated when Cp associates with 1 or 2 regulatory particles Rp on one or both sides of alpha rings. These regulatory particles include 19S proteasome complexes, 11S proteasome complex, etc. Following the Cp-Rp association, the conformation of certain alpha subunits will change and consequently cause the opening of substrate entrance gate. Besides Rp, the 20S proteasomes can also be effectively activated by other mild chemical treatments such as exposure to low levels of sodium dodecyl sulfate SDS or in P-14. As a component of alpha ring, proteasome subunit alpha type -6 contributes to the formation of heptameric alpha rings and substrate entrance gate. The eukaryotic proteasome recognized degradable proteins, including damaged proteins for protein quality control purpose or key regulatory protein components for dynamic biological processes. An essential function of a modified proteasome, the immunoproteasome, is the processing of class I MHC peptides. Clinical significance. The proteasome and its subunits are of clinical significance for at least 2 reasons. 1, a compromised complex assembly or a dysfunctional proteasome can be associated with the underlying pathophysiology of specific diseases, and 2, they can be exploited as drug targets for therapeutic interventions. More recently, more effort has been made to consider the proteasome for the development of novel diagnostic markers and strategies. An improved and comprehensive understanding of the pathophysiology of the proteasome should lead to clinical applications in the future. The proteasomes form a pivotal component for the ubiquitin proteasome system (UPS) and corresponding cellular protein quality control (PQC). Protein ubiquitination and subsequent proteolysis and degradation by the proteasome are important mechanisms in the regulation of the cell cycle, cell growth and differentiation, gene transcription, signal transduction, and apoptosis. Subsequently, a compromised proteasome complex assembly and function lead to reduced proteolytic activities and the accumulation of damaged or misfolded protein species. Such protein accumulation may contribute to the pathogenesis and phenotypic characteristics in neurodegenerative diseases, cardiovascular diseases, inflammatory responses, and autoimmune diseases, and systemic DNA damage responses leading to malignancies. Several experimental and clinical studies have indicated that aberrations and deregulations of the UPS contribute to the pathogenesis of several neurodegenerative and myodegenerative disorders, including Alzheimer's disease, Parkinson's disease and Pick's disease, amyotrophic lateral sclerosis (ALS), Huntington's disease, Creutzfeldt-Jakob disease, and motor. As part of the ubiquitin proteasome system UPS, the proteasome maintains cardiac protein homeostasis and thus plays a significant role in cardiac ischemic injury, ventricular hypertrophy, and heart failure. Additionally, evidence is accumulating that the UPS plays an essential role in malignant transformation. UPS proteolysis plays a major role in responses of cancer cells to stimulatory signals that are critical for the development of cancer. Accordingly, gene expression by degradation of transcription factors, such as 53, c-Jun, c-Fos, NFKB, c-Myc, HIF-1A, MA-TATWO, STAT3, sterol-regulated element-binding proteins, and androgen receptors are all controlled by the UPS and thus involved in the development. Moreover, the UPS regulates the degradation of tumor suppressor gene products such as adenomatous polyposis coli APC in colorectal cancer, retinoblastoma RB, and von Hippel-Lindau tumor suppressor VHL, as well as a number of proto-oncogenes RAF, MYC, MYB, REL, SRC, MOS, ABL. The UPS is also involved in the regulation of inflammatory responses. This activity is usually attributed to the role of proteasomes in the activation of NFKB, which further regulates the expression of pro-inflammatory cytokines such as TNF-alBIL-8, adhesion molecules ICAM-1, VCAM-1, P-selectin, and prostaglandins and nitric oxide known. Additionally, the UPS also plays a role in inflammatory responses as regulators of leukocyte proliferation, mainly through proteolysis of cyclines and the degradation of CDK inhibitors. Lastly, autoimmune disease patients with SLE, Sjogren's syndrome, and rheumatoid arthritis predominantly exhibit circulating proteasomes, which can be applied as clinical biomarkers. PSMA6 has been implicated to be involved in the pathogenesis of ankylosing spondylitis, as it may therefore be a potential biomarker in this autoimmune disease. The same study exploring has also suggested that RPL17, MRPL22, PSMA4 in addition to PSMA6 are involved in the pathogenesis of as and may be potential biomarkers for clinical application as well. Interactions. PSMA6 has been shown to interact with PL-CONE and PSMA3.